Joint stock company International Research and Production Holding Phytochemistry. Bidding for project of team of junior research scientists. Innovative technology of manufacturing of sausalin antiparasitic drug. Project manager, candidate of pharmaceutical sciences A.N. Jabayeva. Project manager is a young scientist in field of technology of drug. Candidate of pharmaceutical sciences Anar Jabayeva, along with her group, consists of highly skilled specialists. Candidate of pharmaceutical sciences Ilya Khabarov. Candidate for a doctor's degree Aygirim Adikenova. Candidates for master's degree Aidar Dairov, Didar Baltash, Aygirim Mukazhanova. The commercialization of research scientific work will be conducted by Yekaterina Yukhnevich, PhD. Participants of project are young scientists up to 35-year-old. Business partner of project is Karaganda Pharmaceutical Plant, which experience in production of original drug based on the raw materials is more than 12 years. Plant has modern material and technical resources allowing the introduction innovative technologies into production of original drugs. Parasitic invasion is global and complicated medical and socio-economic problem in Kazakhstan because helminths and protists have significant influence on public health and economy, increases the number of the diseases and leads to disablement. Currently, prevailing demographic, social, ethnic, environmental situation and the influence of chemical and pharmacological effects on the body of people led to decreased immunity and protective function of hepatobiliary system. In this regard, the rise of parasitic diseases including giardiasis and ophthalmophthisis can be reported. International Research and Production Holding Phytochemistry is one of the leading scientific centers in the Republic in the field of phytochemical research of raw material and creating of original drugs based on them. It has unique infrastructure which involves complex technological development plan of domestic phytopreparations, from the search of biological active substances, plant cultivation and its processing to the organization of pilot production of finished dosage form of phytopreparations. The original drug Sausalin that has lambliasis and antiopistarchasis activities is based on the sesquiterpene lactones of Sausalin salsa, pel, spring, and was developed in the holding Phytochemistry. The active component of Sausalin original antiparasitic drug is cyanopicrine. The concept of the project is that there will be organized area for production of original domestic antiparasitic drug Sausalin in accordance with international standards of GMP. The technology for obtaining of drug Sausalin substances, which is dry extract of Sausalin salsa pel spring, some of sesquiterpene lactones, was developed in recognition of requirements of GMP and consists of following stages. Raw material of Sausalin salsa pel spring is extracted using a solvent system ethyl acetate, ethanol. Obtained extract is evaporated using rotary evaporator pilot valve and undergo aqueous alcoholic treatment for purification from ballast substance. Purified extract is dried using the sublimation drying on sublimator. Finally, dry extract of Sausalin salsa pel spring is obtained that is amorphous hygroscopic powder of olive brown to brown color with specific odor and bitter taste. For obtaining tablets and capsules of drug Sausalin in regard of hyperhydroscopicity of substances, the following technology for obtaining of granules was developed. A V-type mixer is loaded by substance and excipients, thoroughly stirred, and then wet granulation step is carried out. These briquettes are subjected to two-stage wiping on the granulator. The resulting granules are dried and dusted with a V-shaped mixer. Powdered granules are used in the manufacture of tablets and capsules Sausalin. All stages of manufacturing of drug are controlled by using of modern physico-chemical and microbiological method of analysis, UV and IR spectroscopy, high-performance liquid chromatography. Carried out at the Department of Infectious Diseases, Medical University of Astana and Karaganda State Medical University. Clinical trials of Sausalin have confirmed the effectiveness of therapy treatment of diseases caused by parasites. In particular, the drug Sausalin has been proven as effective drug for the treatment of giardiasis and ophthalmophthisis. Therapeutic effectiveness was controlled by two main state medical universities. The business idea of proposed project includes organization of activities for the production of the drug, which main advantages are as follows. Effectiveness of new drug. On the basis of clinical studies, there were received convincing results of efficiency of treatment of diseases caused by parasites. In particular, Sausalin has proven effective drug for the treatment of Opisthoriasis giardiasis. None of the competing drugs have such a broad spectrum of activity. 2. The safety of original drug. The drug has high benefit-risk profile. The only limitation for application is idiosyncrasy of the components of the drug. 3. Availability of drug. On the basis of pharmacoeconomic analysis of the results, it was revealed that out of all of used medicine competitors and biologically active additive Trochatka Avelar, Sausalin may be the comparative economically justifiable for the treatment of parathosis. 4. Plant base for the production of drug. Sausalin is an original drug produced on the basis of domestic raw material plant. The expected results of project are: organization of area for the production of new antiparasitic drug Sausalin. Taking into account its clinical economical advantages, drug Sausalin has the potential to become a competitor to the drug biltricide and biologically active additive Trochatka Avelar. Sales can reach 9,500 units according to the number of patients with giardiasis and Opisthoriasis per 100,000 of population per year according to data of 2014. Signing an agreement with LLP Caraganta Pharmaceutical Plant on obtaining of exclusive license to use the Eurasian patent for a method of Sausalin production.